So with just the last few minutes for today, I want to briefly address how we can use PCR products for cloning. So in the previous video, I told you a little bit about how PCR works, where the broad idea is that we can amplify a gene of interest. But how do we make sure that we can amplify that gene of interest and then put it into our vector of choice, especially in that multiple cloning site? The way that we do that is actually pretty straightforward. When we are ordering our primers, so the red and the blue represent the primers. When we're ordering the primers from the company that makes them, we actually engineer the primers to be just a little bit longer than we need. And in that extra length, that's where we add restriction enzyme recognition sites. So for example, for EcoR1, remember the restriction enzyme recognition site is 6 base pairs. So a primer, this might be anywhere from 18 to 22 base pairs. That's pretty standard for designing a primer. So we would just add the extra 6 for, let's say, EcoR1 right here, and then maybe a different enzyme, let's say BAM-H1 right here. And as this piece gets amplified and amplified and amplified and amplified, you start to generate PCR fragments or PCR products that have the desired restriction enzyme recognition site incorporated. And that's really cool because it then allows you to go ahead and cut that fragment with the same restriction enzymes that you would use to cut your vector so that you can do your cloning. So again, the final product will have those recognition sites that you want. Another really cool modification to PCR, so standard PCR is what we talked about in the previous video. We can modify PCR to use our products for cloning by adding restriction recognition sites. The last modification to PCR that I want to introduce for this section is the idea that we can actually use PCR to help us figure out which amino acids could be important for protein function. So how do we do that? Well, we use a process called site-directed mutagenesis. Site-directed mutagenesis. Let's say this is our start codon, here's our stop codon, and we are interested in changing this particular amino acid. What we do is we actually design primers, right, to amplify this region, and we need to change a nucleotide. Maybe 1, maybe 2, to swap out our amino acid. Remember, there are some amino acids where multiple codons, so multiple 3 nucleotide reading frames will give us the same amino acid. So in that case, maybe we'd have to change out 2 nucleotides in order to swap out the amino acid. But by having a primer here that will amplify this part, where we have, in our primer, we put the desired sequence. So this is the standard sequence, here's our desired sequence. With that primer that has our desired change, right, when it binds, first we'll have, here they're showing the mutation of interest, right? The primer won't bind right here, but that's okay. That slight mismatch is totally fine. But as we start to amplify over and over and over again, that change gets incorporated every time this piece is amplified. So eventually, we end up with our mutant. And that can help us to identify protein function. So it's a really cool application of PCR. As we saw in the video, PCR and cloning became more common starting in the early 1970s to the 1980s. And so in the early 1970s, we were working with restriction enzymes and plasmids that allowed us to do gene cloning. And this was really described as a new era in molecular genetics. There was so much information that scientists were finally able to generate, but that opened up a host of new questions. And so what we'll talk about the next time we have a lecture class, because remember our exam is next week, so the week after that we'll have our next lecture class, is how understanding cloning, understanding PCR began to allow us to develop technologies to sequence DNA. So how did we go from PCR to sequencing? And so that's going to be our next step when we talk about new technologies.